So we will continue our discussion on proteins, but right now we will have the formal discussion, which means we will be able to know how proteins really form themselves, starting from the attachment of one amino acid to another until such time that we have a long chain of amino acids or polypeptides, how proteins are formed from polypeptides, what were the changes, what were the processes. We will also discuss about the classification of proteins based on their shapes and functions. We also have the post-translational modifications. These are modifications or changes that are made on proteins so that the proteins will have their specific functions, or so that the proteins will be activated. We will also talk about the examples of proteins. So what are, the usual proteins that you hear every day, usual proteins that you can easily think of. So proteins are biochemical compounds consisting of one or more polypeptides, typically folded into a globular or fibrous form, facilitating a biological function. So when we say proteins, proteins or fibrous proteins, these are actually classifications of proteins based on their shape. So there are proteins that are of globular in shape. There are proteins that are of fibrous in shape. And depending on the shape of the proteins, we would be able to know what would be the function of the proteins, what are their roles in the biochemical process. Polypeptide, I guess this has been given to you in the previous video. This is a single linear polymer chain of amino acids bonded together by peptide bonds between the carboxyl and amino groups of adjacent amino acid residues. So this is just a review from the previous video or lecture video that, and I guess most of you have already accessed. So a polypeptide is basically, amino acids, a lot of amino acids that are bonded together to form a very long chain of amino acids. Very long chain of amino acids would be equivalent to polypeptide. And polypeptides also attach together in order to form a longer version of polypeptide. Then the longer version of polypeptide will then need to fold in order to produce the so-called proteins. The sequence of amino acids in a protein is defined by the sequence of a gene, which is coded in the genetic code. Actually, I don't know if I've already mentioned this one, but basically proteins are produced are produced starting from, codes that are being carried by mRNA, your messenger RNA. So basically you have the DNA. First, you have the DNA, right? In sorry, my graphic tablet is not working really well, probably because there's still, another PowerPoint presentation that I am currently converting into video. So I guess that's why it's lagging. But anyway, we have the DNA. I'm just using the built-in mouse of my laptop. We have the DNA from the nucleus of the cell. So the DNA is basically the template. So this is the DNA, for example. Then from the DNA, we need to form what we call a copy of it in the form of mRNA or the messenger RNA. The messenger RNA will then need to go out of the nucleus and then travel until it reaches the ribosomes. In the ribosomes, the mRNA will be decoded because your mRNA carries codes. Remember your CG80, your cytosine, guanine, adenine, and tyrosine? They're called your bases, right? Or bases, nitrogenous bases. So anyway, I'll just give you a review on that when we reach the topic on nucleic acids. But anyway, I'll just give you a gist of what happens during decoding. Again, your mRNA carries codes. So a code basically contains three set. The code is actually a set of nucleobases. So may tatlo kada ka nucleobases. Whatever the combination. Ang tatlo na nucleobases will be equivalent to one code. Example, this one, wala na da T sa mRNA ha, uracil. Thymine is the nucleobase that can only be seen in the DNA. Ang uracilya, amo na iya ang makita nimo sa RNA. So that's why, gin-erase ko ang T, ginislan ko U. Kaya wala tayo T nga nucleobase sa mRNA. So anyway, for example, ACU codes for, example lang niya, ACU codes for isoleucine. For example, leucine na lang LEU. So that specific code, ACU, again, codes for LEU. So, amo ni ang code na gina-carry, for example, ni mRNA. Code B is leucine D, another tatlo decanucleobases ga code for methionine, another tatlo decanucleobases ga code for alanine. So, ang code na kinanglan i-decipher, i-decode. So, you have the tRNA. Sorry for my drawing. Sige lang. Let me erase this one first. So, example, this is the mRNA. Tapos mo ning code ya. Tatlo ni siya ka-code ha. Tatlo ni siya nucleobases de. Tatlo man de. Next, we have the tRNA here. So, tRNA would look like this one. Doka clover leaf na siya. And then basically, may-ari di siya nga decoder. Ang decoder yadi is actually the pair man or pares man sang code nga ara sa mRNA. So for example, ang code is ACU sa mRNA. Dapat ang makadecode siya would be pares ni AU, pares ni CG, pares ni UA. So if this is the code found on the mRNA, dapat ang makadecode siya is UGA, which is found sa tRNA nimo. Now, once gani, ang decoder sa tRNA, kagang code sa mRNA, mag-swak, pwede na dayon ma-materialize ang imo nga amino acid. For example, leucine. And then the tRNA will just move to another naman nga code sa mRNA. Then, ipares yan na daang decoder for that specific code to produce another naman na amino acid. The first amino acid, leucine, will need to be attached to another amino acid that will be produced, to the second amino acid that will be produced. And the same process will go on and go on until such time nga magdamo ang amino acids nga na-produce. Kag ang damo ng amino acids are constantly being attached to each other. Actually, I just explained to you how protein translation happens. But anyway, no need to fuss. I just showed you how, the meaning of the first, part here of the discussion, or meaning sa first bullet. The sequence of amino acids, again, in a protein is defined by the sequence of a gene, which is encoded in the genetic code. So I just showed you how is that being possible. In general, the genetic code specifies 20 standard amino acids because only 20 amino acids have, corresponding codes nga makitaan sa aton nga DNA kag pwede makopy sa mRNA. However, in certain organisms, the genetic code can include selenocysteine, which is still an amino acid, and in certain archaea, we have pyrrolysine. So in humans naman niya and animals, we have 20 standard amino acids. Some galing na organisms, especially the archaeans, they have what we call other amino acids or genetic codes for amino acids that are, different from the 20 standard amino acids. There are organisms that can actually code for selenocysteine and pyrrolysine. And take note, guys, these two amino acids are not found in our body. Wala niya sa aton lawas lawas ng tao, wala na sa lawas ng animals. Shortly after or even during synthesis, the residues in a protein are often chemically modified by post-translational modification, which alters the physical and chemical properties, folding, stability, activity, and ultimately the function of the proteins. This is what I've mentioned in the beginning of our video. Post-translational modification is basically a process wherein your proteins are being edited, right? The term is edited. They are further changed or edited so that the chemical properties, physical properties of proteins are enhanced, and so the functions of the proteins will also be established. Monomeric protein basically has one peptide chain present. So basically, amo ni mga proteins nga mga gagbay masyado nga proteins. Monomeric. When we say multimeric proteins, basically they have two or more, more than two peptide chains connected to one another. Mono, which means one. Multi, which means multiple or greater than one or greater than two. Yep. So a multimeric protein is also composed of monomeric protein. The protein subunit is the monomeric protein. So the multimeric protein is just a combination of monomeric proteins being bound together. Yep. And the monomeric protein is composed of amino acid residues. So ang pinaka-basic would be the amino acid residues. The amino acid residues are composed of 20 amino acids, 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 amino acid residues would form your protein subunit, which is the monomeric protein, and more monomeric proteins bonded together will form your multimeric proteins. So this is how we visualize protein hydrolysis. So as you can see, we have here an example of peptide. So we will just have to observe in a smaller scale na lang danay, not the whole protein itself, kaya medyo complicated. Let's just have a very easy example. So let's say peptide ni siya, which is true, no, because we have here one amino acid. Let me show you. This is one amino acid, right? Correct man. You have the alpha carbon atom, we have the carboxyl group, we have the hydrogen here, the side chain, and of course the amino group. Next, we have another amino acid here, the alpha carbon atom, hydrogen, side chain, carbonyl group. Carbonyl na siya guys ha, because kulang na siya sang OH. So carbonyl, and then we have the amino group. Next, we have another amino acid here. We have the alpha carbon atom, hydrogen, side chain, and your carbonyl. Now, in hydrolysis, there is actually a usage of water. So we use a water molecule in order to break the proteins. Aside from water molecule, we are also using chemicals, or for example, acid, base, or we're also applying heat in order to somehow initiate the breakage of the peptide bond. So the peptide bond again is the one that is being, encircled here by the blue circle here. Yes, the C double bond O and H, that's your peptide bond. That is where your amino acids are being linked together. So the goal of protein hydrolysis is to target the peptide bond, break the peptide bond to release your amino acids or peptides, smaller peptides. Again, what do we use during protein hydrolysis would be 1. Water, H2O. We may also be used some chemicals such as acid, base, as well as heat. And there are also methods which use physical force. So there are chemical as well as physical methods in order to initiate protein hydrolysis. But basically, protein hydrolysis and all other types of hydrolysis make use of water molecules. So with the use of chemicals or physical method plus the water molecule, we are now able to break the peptide bond, releasing now this amino acid. Ma-release na siya. And then this one will also be released. Notice that this is not an amino acid but still a peptide because this is still a combination of two amino acids, correct? Ari isa na siya ka amino acid, ari isa man ka amino acid nga nakatapik pa. That's why peptide pa siya. So in protein hydrolysis, as I've mentioned, the result could be amino acids laban amino acids. Nga pwede man may sakot nga peptides. Depende kung gaano ka-extensive ang method ginagamit for the protein hydrolysis. Now, in protein hydrolysis, notice that the amino group here, previously in this specific peptide, has now gained one hydrogen atom. From NH, it has become NH2. And the peptide here is just having the C double bond O. But after hydrolysis, it has, gained its carboxylic group structure. Nanging C double bond OOH na siya. Where did we get the OH here and the H here? Actually, the OH or hydroxyl and the hydrogen here came from the water molecule that was used in the reaction. One hydrogen was reacted with the amino group of this molecule, whereas the OH here from the water molecule has reacted with this specific molecule. So basically, your protein hydrolysis is the opposite of dehydration reaction or condensation reaction. ON. Dehydration or condensation reaction. Remember that in the first lecture video, condensation. In the first video, I've mentioned these two. Dehydration reaction or condensation reaction is basically a process wherein, proteins are being made by combining amino acids with the production of water. Remember, in dehydration or condensation reaction, may ginaliberate nga water molecule. Sulit naman sa protein hydrolysis. When we break down proteins, we are actually using water. In dehydration process, we are releasing water. Take note of that. Another visual representation of protein hydrolysis. Kindly disregard this one because this is not included in our discussion. Let's just, focus our attention here. For example, this would be your proteins. And depending on the type of protein hydrolysis or the method that we use for protein hydrolysis, we would be able to get different types of products. For the extensive one, which means grabe gid ang method ginagamit probably we use a very strong acid or a very strong base in order to somehow break down proteins, or probably we use proteolytic enzymes because proteolytic enzymes really are very effective in breaking down proteins into amino acids or smaller subunits. So as you can see here, if this is the original protein, after extensive hydrolysis, we can now only have smaller pieces of peptides. See, they are really small. And probably we might be able to somehow have, a couple of amino acids there. So not just peptides, but also amino acids probably would be the products of extensive hydrolysis. For the partial hydrolysis, we might be able to have, peptides still, but but we can still have long peptides or polypeptides, such as this one. We might be also have, smaller types of peptides, dipeptides, tripeptides, but rarely amino acids. Rare na lang diyang amino acids sa partial hydrolysis. For the mild hydrolysis, since this is just a mild hydrolysis, most probably ang makuha tadi would be polypeptides. Polypeptides. Right. Kaya wala masyado na breakdown ang mga peptides dira. Kaya mild hydrolysis man lang. So, just 1, 2, or 3 peptides lang siguro ang naguba. So, in that case, well, we will expect that the products that we will have would be polypeptides lang. Utod-utod nga polypeptides. For the partial hydrolysis, we will have polypeptides also, but we might be also having oligopeptides, dipeptides, tripeptides, or smaller types of peptides. Not so much with amino acid. For the extensive hydrolysis, yes, pwede kita makakita amino acids plus oligopeptides, dipeptides, tripeptides. Polypeptides, not so much the extensive hydrolysis, because again, this is already an extensive one, which means a lot of, peptides have already been broken. Now let's talk about some, peptides that are basically being used in the different biochemical processes here in our body. For example, we have some peptide hormones like your oxytocin and vasopressin, or ADH, or antidiuretic hormone. Again, vasopressin or antidiuretic hormone. For the oxytocin, it regulates uterine contractions and lactation. So basically, high oxytocin level ang may ara sa pregnant women, most especially during labor. For the vasopressin, ADH, all of us have this one, ang among nga hormone. Antidiuretic hormone, which means this is the hormone that we need in order to not lose so much water through urination. For example, if we are dehydrated, the body will increase the level of antidiuretic hormone, which means the antidiuretic hormone will stop us from urinating, will stop our body from releasing water into the kidneys. So that's why if you are dehydrated, dutay man ang imo ginapangihi. And that is thank you. Pa-thank you ta kay ADH, antidiuretic hormone. This regulates the excretion of water by kidneys. And yes, this hormone basically affects the blood pressure. And actually, the blood pressure also affects the release of the vasopressin or ADH, antidiuretic hormone. Why? Because if you have low BP, that means you have low level of liquid in your blood. Right? Low level of liquid in your blood, which means low level imo plasma, which means blood volume mo is medyo dutay man. Low level of blood volume will basically be detected by organs of our body. So the organs of our body will send signals to the brain, hey, we are receiving low amount of blood here, probably because low ang blood pressure. The body will then release ADH, antidiuretic hormone, because the body will think the reason for the low BP is because low ang plasma, low ang liquid portion of the blood. So the body will release ADH, antidiuretic hormone, so that because of antidiuretic hormone or through antidiuretic hormone, hindi masyado mag-release ang tubig ang aton nga kidneys. More water will be retained in our body so that hindi gid masyado akag hindi mag-further low or reduce ang BP naton. See, that's the magic of your hormones. But anyway, more discussion on vasopressin or antidiuretic hormone when you get to CC or clinical chemistry. And I tell you, for me, it's just my opinion, but I hope someday you will also, figure it out yourselves how how fun it is to learn about hormones. Sige lang guys, kaya nyo na. These hormones, oxytocin and vasopressin, are known as nonapeptides, which means peptides sila, which are composed of 9 amino acids. Nona, which means 9. May 9 ka-amino acids that are bonded together to form one peptide. So, nonapeptide siya. And we have another set of peptides, but this time they are neurotransmitters. When we say neurotransmitters, they are chemical messengers that basically transmit a signal from one neuron to another, or from one neuron to the target cell. For example, we have the enkephalins. They are pentapeptide neurotransmitters. Penta, which means 5. That means a pentapeptide has 5 amino acid residues. Enkephalins reduce pain. Right? So be thankful to enkephalins. They will be able to reduce the pain that you are feeling. Example of enkephalins would be your methenkephalins, which is basically composed of tyrosine, glycine, phenylalanine, methionine sequence. We have leuenkephalin, which is composed of tyrosine, glycine, phenylalanine, leucine, amino acid sequence. We have an antioxidant peptide, glutathione, which you can basically find in your beauty products, in your soap, in your lotion, in the tablets that you are taking, whitening tablets that you're taking. Glutathione is basically composed of glutamic acid, cysteine, and glycine amino acid residues. It is a tripeptide. Tri, which means this is a peptide that is composed of 3 amino acid residues. Halata man, tatlo man ka amino acid rio ang ari sa kay glutathione. Glutathione is a regulator of oxidation reduction reactions. That's why it's also good to take glutathione tablets. Because basically they help, the glutathione itself helps in the oxidation reaction of the body. If we're stressed, there are a lot of free radicals that are continuously produced by our cells, and we need to deal with all of those radicals or free radicals that are inside of our body. Free radicals such as superoxides can actually kill our cells or damage or worse kill our cells. Now, if we take glutathione, vitamin E, vitamin C, which are all good for oxidation reduction reactions, we will be able to somehow lessen the free radicals that we have in our body. Next, we have the protein classification. Proteins can be classified as simple or complex proteins. For simple proteins, they are just pure amino acids. Proteins that are just, that are composed only of amino acids. No other organic molecules are present in the structure of protein. For the conjugated protein, it has one or more non-amino acid entities, which means we have the amino acid part, and some part of the proteins are actually lipid parts. Some parts of the proteins are actually carbohydrate parts. Prosthetic groups or cofactors. There are non-amino acid or non-peptide entities in a conjugated protein structure. So later we will see the different, examples of simple protein, conjugated protein, and prosthetic groups or cofactors. So you have here the conjugated protein classification. Lipoproteins, which basically has prosthetic group. Lipids, which means that is a protein that has a lipid portion or may parts sa iyang body nga lipids. Glycoproteins, from the prefix glyco, may carbohydrate siya na content. Lipo, lipids, glyco, carbohydrates. Metalloproteins. The protein basically has a metal ion in it. Hemoproteins basically have a heme unit, just like your hemoglobin. It is a hemoprotein because it carries the heme unit. Next, phosphoproteins basically has a phosphate group, phosphate. Nucleoproteins, proteins that have nucleic acid content. Now we have examples of your lipoproteins. We have the low-density lipoprotein or the LDL and high-density lipoprotein or HDL. LDL is dubbed as the bad cholesterol or bad lipoprotein, whereas HDL is dubbed as the good cholesterol or good lipoprotein. Why is it that your LDL is called as bad cholesterol? Because your LDL is the one that carries your cholesterol to your arteries. So if you have high content of bad cholesterol in your body, which means you have high amount of low density lipoprotein in your body, that means you will also have a high probability of increased deposition deposition of cholesterol in your arteries. That also means that you have high probability of developing arteriosclerosis. High-density lipoprotein, on the other hand, is called as good cholesterol. Why? Because it carries your cholesterol from your arteries going back to your liver. So in your liver, this is where your cholesterol is being reprocessed or processed further. Again, good cholesterol is your HDL because it basically carries all cholesterol from the arteries going back to the liver. Here are the examples of your glycoproteins. You have number one, collagen, which you already know is a structural protein. Right? Collagen is there in order to provide support to the structure of the body. Then we have mucin, which is a glycoprotein constituent of your mucus. Mucus is, again, is a good lubricant of your um, of what your nasal cavity, and lubricant also in your intestines. Transferrin Transferrin is basically a protein which binds and transports the iron. Again, it binds and transports iron in blood serum. So the movement of iron in your body depends on the transferrin. We have immunoglobulins. Well, immunoglobulins are proteins which are basically produced by the body in response to the invasion of a foreign substance in your body. Uh, for example, if a virus, if a fungus, or let's say a bacterium gets inside of your body, since they are considered as foreign materials, your body will then recognize them as so, as foreign materials. Then in response, the body will get rid of those foreign materials. Ridding of the foreign materials would include the production of immunoglobulins, also known as antibodies. Immunoglobulins are the ones that will neutralize the antigens or foreign materials that got inside of your body, so that those foreign materials won't be able to cause any harm in your body. The virus will be inactivated, the bacterium will be inactivated, the fungus will also be inactivated, and all those stuff are inactivated with the use of immunoglobulins. We have histocompatibility antigens. Your histocompatibility antigens are actually receptors on the cell surface of donated organs that are known to give that immunological response in some organ transplants. So, immunohistocompatibility antigens are what we usually try to check during, assessing if it is possible for the donor to donate a specific organ to the recipient. So the donor and recipient has to be matched. Has to be matched or cross-matched with each other. Their histocompatibility antigen should also be matched, so that there would be no so that there would be low probability of organ rejection. So, amo ni ang mga antigens nga gina-check sa organ sang donor. The antigen should be, compatible with that of the body of the recipient of the organ. Then we have the metalloproteins. We have hemoglobin. It is a metalloprotein because your hemoglobin has iron in it. Iron in the form of ferrous Fe2+. And I guess you already know the function of hemoglobin. It is actually the protein pigment of your blood, which basically carries the oxygen molecules. Calmodulin um, is also a metalloprotein that um, has a job of detecting or sensing the rise and fall on the calcium concentration. Um, yep, a calmodulin is the one that senses or monitors the concentration of calcium. Uh, because we all know that calcium is really needed for the normal muscular contraction. So it's the job of the calmodulin to send signal whenever calcium is lacking in the skeletal muscle. Then we have the troponin, which is basically a metalloprotein that when complexed with actin or tropomyosin, which are other proteins by the way, they basically produce muscular forms in your heart. So troponin is a metalloprotein that you can find in your heart, more specifically in your heart muscle. With the use of troponin, your heart muscles were able to produce or exert that specific force enough to somehow have the pumping of the blood get going. So whenever a person gets a heart attack, it usually it is the troponin levels that are being monitored. If the troponin levels are high, that means that person indeed suffered from a heart attack. Because only when a person had a heart attack, that is also the time when troponin levels will increase in the blood. So I will let Miss Jem to further explain the significance of high level of troponin in the blood of the patient. Next we have the hemoproteins. We also that is hemoglobin, sorry for the typo. We have hemoglobin because hemoglobin contains a heme unit. Myoglobin also contains heme unit. Myoglobin is quite different from hemoglobin because myoglobin is the one that stores oxygen in the muscle. It does not transport oxygen unlike hemoglobin. Catalase is an enzyme that is found in the liver. The function of the catalase in the liver is in breaking down harmful hydrogen peroxide, H2O2, and it breaks down hydrogen peroxide into oxygen and water. Again, catalase is the one that breaks down hydrogen peroxide into oxygen and water, and it is an enzyme found in the liver. Cytochrome. Cytochrome is basically an electron carrier. Sige lang, I will just introduce you the electron carriers once we reach our discussion on metabolism, most especially in electron transport chain. So cytochrome is very important in metabolism. That's it. Phosphoproteins, we have casein, which is the protein component of your milk. We have stathurine, which is a phosphoprotein found in your saliva. Lastly would be nucleoproteins. We have telomerase and histones. Telomerase is a protein that modifies the nucleic acid. Sige lang, I will just let Sir Jericho introduce you to telomerase. Histones I guess you are already, familiar with histones. Histones is where are where your chromatins are being packaged, right? Remember, there is this specific structure wherein chromatins are being wind to. So histones na siya. And um, it is mentioned by our professor in our master's program in CDU. And it is also mentioned in some articles and books, molecular biology books and biochemistry books, that the histones actually are equivalent to the age of a person, right? As well as it also determines how long we are going to live and how long we are going to stay young, right? So if we have a shorter histone, then that means we will only stay young in a short amount of time. We will grow old very fast. Yep. So that that was, the context that we found in some books, in some, articles, as well as, the context that were given by our teacher. Histones actually uh, carry the program of your body. I mean, carry the program of when your body will, cease in functioning. Proteins can also work together to achieve a particular function, and they often associated to form stable protein complexes. That is true. Guys, please take note that even though our organic molecules, proteins, carbohydrates, lipids, and nucleic acids are different in terms of their structures, but then again, um, at some point, these organic molecules um, can also work together in order to play a major or important role in biochemical processes in the body. So it would be possible that without carbohydrates, biochemical processes will cease or will stop. Without proteins, no biochemical processes will occur. Without nucleic acids, everything will shut down. Also, without lipids, everything will not work. Proteins serve in many roles, such as enzymes, hormones, receptors, antibodies, structural components, transporters of other compounds, and contractile elements in your muscles, such as your actin and myosin. Enzymes, yep. Some metalloproteins are also enzymes. Hormones, well, you already encountered your oxytocin and what else? Oxytocin and your vasopressin or ADH. Receptors, yes, your glycoproteins are also receptors. Antibodies, as I've mentioned, your immunoglobulins. Um, Yep, your immunoglobulins. Then we have the structural components. You have keratin. It is a structural component. Collagen. It is a structural component. Transporters of other compounds. You have transferrin. It transports iron. Hemoglobin can be also considered as a transporter because it basically transports oxygen. And contractile elements, again, you have your actin and your myosin. Let's have protein classification based on structure. So we have the primary, secondary, and tertiary structures of your proteins. Primary is basically the level 1 structure of your proteins. It is consisted of amino acid sequence along the chain. So, amino acid lang gid siya. Amino acid lang gid siya. Secondary structure would involve alpha helices, beta sheets, and other folding patterns. So from primary structure with only amino acid sequence, there will be what we call the start of folding. That's secondary structure. Next, for the tertiary structure, there's what we call three-dimensional conformation of proteins. So there is already the height, width, and length of your protein shape or protein structure, which involves your electrostatic and hydrophobic interactions plus hydrogen and disulfide bonds. So from simple amino acid sequence, if you just add a little bit of folding, that is now your secondary structure. From secondary structure, if there are more interactions involved plus bondings that will be involved, it is possible that we could form a three-dimensional structure like that, forming now the tertiary structure of your protein. That is why studying proteins is really hard, because we don't know what we cannot really predict how would proteins fold. We cannot predict in what manner the proteins will fold. How would they be able? How would we be able to predict the final three-dimensional structure of the proteins? Lastly, we have the quaternary structure, which refers to the interaction of one or more subunits to form a protein. So basically, you have folded protein here, and another folded protein here, and another folded protein here to form your quaternary structure. Scribble lang guys. Pero, yeah, basically, ako nang kakatabo sa proteins nimo. There were a lot of twists and turns. There are a lot of interactions, bondings that occur. And then lastly, you have the quaternary structure wherein three-dimensional structures bind together in order to form a bigger structure. Let's have the general aspects of protein structure. The linear sequence of amino acid residues in a polypeptide chain determines the three-dimensional configuration of a protein, and the structure of a protein determines its function. Right. So the linear sequence of amino acid residues determines the three-dimensional configuration of the protein. Actually, yes, it kind of makes sense. Why? Because remember, there are different types of amino acids. Amino acids that hate water, amino acids that love water. So based on the properties of amino acids, the amino acid chain will be able to move accordingly, will be able to fold accordingly. Right? There are amino acids that are compatible together to form one interaction. Pwede sila ka-form bonding, for example, hydrogen bonding, salt bridges, whatsoever. And because of the capability of amino acids to form bonds or interactions with other amino acids, pwede sila ka-form some different foldings. For example, this amino acid can be able to form interaction with this amino acid. So, ma-fold siya. And then the next amino acid will be able to form interaction with this amino acid. So, ma-fold naman di siya apart. Then, for example, this amino acid is hydrophobic. So, hydrophobic siya dapat mapilo siya away from water. And this amino acid sequence loves water. So, dira sila mapagwa sila. Then, the same process will go on. Next, the structure of a protein determines its function, correct? If there is any alteration or changes in the structure of proteins, pwede nga hindi na maka-perform sa function ng protein. Remember, I've mentioned many times before that the function of proteins is dependent on the shape of the protein. If the shape of the protein changes, that means the protein will have this means that the protein might not be able to perform its biological function because again, guba ang iya shape. And there might be important structures of the protein nga kinahanglan sa biochemical process nga hindi na magamit sa biochemical process. Let's start with the primary structure of the proteins. Again, recall it is the sequence of amino acids along the polypeptide chain. By convention, the sequence is written from left to right, starting with the N-terminal amino acid. Because there are no dissociable protons in peptide bonds, the charges on a polypeptide chain are due only to the N-terminal amino group, the C-terminal carboxyl group, and the side chains on amino acid residues. A protein will migrate in an electric field depending on the sum of its charges at a given pH, or the net charge. Positively charged proteins are considered as cations, and they tend to migrate toward the cathode, or the negatively charged side. The negative charged proteins are known as anions, and they tend to migrate toward the anode, or the positively charged field. At the isoelectric pH, the net charge is 0, and the protein does not migrate at all. So I guess we're talking about what we are talking about is the movement of the proteins in the electrophoresis. Guys, electrophoresis is basically a process wherein we let the smaller substances, fragmented proteins to be exact, to migrate from one field to another. And their migration is dependent on their charges, right? If they are positively charged or negatively charged. Those proteins with, zero charge, of course, they won't be able to migrate. They won't be able to migrate either the cathode or anode field. For the primary structure of your protein, you just have the sequence of amino acid. No folding at all. Guys, when I say folding, it's not just normal folding like this. This is not the secondary structure. This is still the primary structure. When I say folding, when we are talking about secondary structure, there is already the involvement of interactions or bondings. Later, I will show you. But basically, when you say primary structure, it is just the chain of amino acid residues. No bonding between neighboring amino acids. Kita mo wala bonding halin diri nga part, pakadto diri, wala bonds. All we have is just one sequence of amino acid. Secondary structure again includes various types of local conformations in which the atoms of the side chains are not involved. So the side chains are not involved yet in the bonding here. Alpha helix is one of the types of secondary structure. Alpha helix is generated when each carbonyl or the C double bond O of a peptide bond forms a hydrogen bond with the NH of a peptide bond, 4 amino acid residues further along the chain. So let's have, an image of that. Right? So we have the alpha helix here. As you can see, first we have the sequence of amino acid. Next, for the secondary structure, there's already the involvement of, yes, bonding to neighboring amino acids, most especially hydrogen bonding to neighboring amino acid. And the hydrogen bonding happens between the carbonyl group, so C double bond O, carbonyl group of one amino acid with the NH or amino group of another amino acid. Yep, that is 4 amino acid away from one another. So we have one amino acid to 3, yes, 4 amino acids. So we have 4 amino acids ang lakang nila before sila nagbonding. Same with this one. Same with this one, nga pair. Same with this. Beta-pleated sheet is another form of secondary structure. There is also a hydrogen bonding involved between the carbonyl group of one amino acid and the amino group of the neighboring amino acid. Beta-pleated sheet. The side chains of the amino acid residues extend outward from the central axis of the rod-like structure because they're not yet involved in any bonding at all. So all of the side chains of the amino acid tend to extend outward of the axis. Gapaguha ang ilay nga direction. The alpha helix is disrupted by proline residues in which the ring imposes geometric constraints. Remember, the side chain of proline connects with the amino group of proline, and by regions in which numerous amino acid residues have charge groups of large, bulky side chains. So we just have some, problems when it comes to prolines and amino acids that basically have bulky side chains. Pag maka-bulky, do kadagko sa nila side chains that basically are of long aliphatic structures or molecules. Laba-laba katama ang side chain. So with the presence of long or bulky side chains and the constricting side chain of proline, there is what we call a restriction man in the formation of your alpha helix. Beta sheets or beta-pleated sheets are formed by the hydrogen bonds between two extended polypeptide chains or between two regions of a single chain that folds back on itself. These interactions are between the carbonyl of one peptide bond and the NH of another. So it was already provided in the previous the last two slides. The chains may run in the same direction or in opposite directions. So again, we have the beta-pleated sheet here, and as you can see, again, there is hydrogen bonding between the carbonyl group of this side and the amino group of this side of the sheet or peptide chain. Right, so here is another orientation of the beta-pleated sheet. The sheets that run in the same direction. Then we have the alpha helix. Right? Notice in your alpha helix, all of the side chains are extending outward. If this is the axis, or the center of the alpha helix, notice that all the side chains are doing is extending outward or away from the central axis. You have here the side chain here. Yep, they tend to, extend outward. Supersecondary structures. Certain folding patterns involving alpha helices and beta sheets are frequently found and include the helix turn helix, the leucine zipper, and zinc finger. Other types of helices or loops and turns can occur that differ from one protein to another. We try to somehow differentiate random coils with that of the secondary structure. When you say random coils, they don't really have yeah, there are no bonding at all, right? No bonding at all. There's no definite shape. Random coils lang yun sila. For the supersecondary structures, surely there is what we call what we call a sure folding of the amino acid sequence, plus there is what we call bondings between neighboring amino acids. As for the random coils, there's no definite shape, right? And there's no bonding at all between neighboring amino acids. So this is one of the examples of secondary or super secondary structure. We have the helix turn helix. Again, this is a helix turn helix. So as you can see, we have one part of the structure here, which is an alpha helix. The alpha helix just a part of the alpha helix, just turned on the same direction, forming another structure here, the same as an alpha helix. There are no certain bonding here. This is just the part wherein amino acids are in sequence. No bonding at all. But coming from this part and here, it's not really shown, but there is really some bonding there, hydrogen bonding between neighboring amino acids. And you have the leucine zipper. Yep, there's some coiling, but the most important thing is there's still what we call bondings between the two coils. Same as this one. And this is the, I guess this is the original, no, this is the original concept. This is the more correct concept of the leucine zipper. Alright, so you have here another example of secondary structure. Yep. You might think that this is just a random coil, but it's not, because there's still what we call, bonding somewhere in this specific structure. The tertiary structure of a protein refers to its overall three-dimensional conformation. It is produced by interactions between amino acid residues that may be located at a considerable distance from each other in the primary sequence of primary sequence of the polypeptide chain. Hydrophobic amino acid residues tend to collect in the interior of globular proteins. We already discussed this one. I've mentioned in the previous lecture video, that all hydrophobic amino acids tend to fold inward. They tend to avoid water. That's why they are only located inside of the protein, most especially inside the globular proteins. They exclude water. They extend away from water, while hydrophilic residues or hydrophilic amino acids tend to move outward, tend to be located outward or outside or on the surface of the proteins. Why? Because they're okay to be interacted with water. The types of interactions between amino acid residues that produce the three-dimensional shape of a protein include hydrophobic interactions, electrostatic interactions, and hydrogen bonds, all of which are non-covalent. Covalent disulfide bonds also occur in the three-dimensional structure of protein. Let's see, this is an example of a three-dimensional structure or tertiary structure of protein. Notice that at this point we have alpha helix here, alpha helices, right? And then, yep, we have super secondary structures here na do hindi natin maintindihan ang iya nga what shape. But then again, we are sure that there are bondings happening there. Yep, we also still have the alpha helix mandere. I think this is the helix turn helix. Yeah. As also in this one, helix turn helix. Right. So there are a couple of secondary structures there bonded together to form another level of structure, some pure protein, which is the tertiary structure. So basically, as what we can see, the tertiary structure of protein is quite more complex compared to the secondary, and much more to the primary structure of proteins. The heme compound is, yeah, the heme compound is a tertiary structure. We have here some foldings, right? Yeah, this is a very long alpha helix going on here. Actually, this is not the heme, sorry. This is the heme portion, but this is one of the subunit of your hemoglobin, sorry. This is one hemoglobin portion or one hemoglobin subunit. Each subunit of hemoglobin contains a heme unit. Right, so this is one part of the hemoglobin lang danay. And as you can see, there is already what we call the tertiary structure. Yeah, yes, still the same, but with just a little different representation, or presentation rather. Wow. So this is a much more complicated tertiary structure. Let's have the quaternary structure. Refers to the spatial arrangement of subunits in a protein that consists of more than one polypeptide chain. The subunits are joined together by the same types of non-covalent interactions that join various segments of a single chain to form its tertiary structure. For example, we have, Arina, we have a hemoglobin. Guys, yes, this is how hemoglobin would look like. And the hemoglobin is actually composed of 4 subunits. We have, hang on, This one, this is one subunit, second subunit, third, and last subunit of hemoglobin. So basically, the hemoglobin is composed of 4 subunits of polypeptide. We have the beta chain here, beta chain, alpha chain. This is also the alpha chain. This is also the beta chain. Right? And they are actually bonded together, man. We have the heme and the iron component of hemoglobin. There. So quaternary structure is the most complicated structure of protein because it is now a combination of tertiary structures being bonded together. So you have here the primary structure, which is just a sequence of amino acid, and then later on, the long chain of amino acid tend to somehow form some coilings or loops, and then foldings. Yep, and then with that, there are also some hydrogen bondings going on here and there. Then more bondings will form, forming now your tertiary structure, which now has its three-dimensional conformation. Yes, may makitaan ka na nga with what do ka length niya, height niya. Then these these tertiary structures will then attach together to form a bigger structure known as your quaternary structure.